All right, we're moving on to chapter 8 of our microbiology text, or Tortora textbook. Now that we know quite a bit about microbial growth, we're going to start looking at some issues of microbial genetics. And we'll find out a little bit about mutations that may occur, ways that bacteria, for example, can share genetic information, which is kind of a scary and difficult issue. There's a little picture of uh, actual plasmid molecules, double-stranded DNA molecules, if you can imagine such a thing being visualized by high-resolution microscopy. All right, so genetics is the study of genes, how they are used to produce proteins, the information for producing proteins, and how it's encoded in the DNA, and how that becomes replicated when a microorganism wants to or is about to proliferate. It needs to replicate or copy its genetic information. A gene is a single unit of genetic information. It's all the information needed to translate into a protein. It's basically the recipe for the amino acid sequence of protein, and plus any regulatory DNA that's needed to control the expression of that gene. A chromosome is the part of the genome where all the genes are assembled. One chromosome in a bacterium contains all the genes that encode all the proteins that the bacterium can make. In a more complex organism, there may be many separate chromosomes, each one of which contains some of the genes for translating some of the proteins in that organism. So again, the genome is all of genetic information in a cell or in an organism. So genomics is the study of the genomes at the molecular level, anything to do with that. But that's not so important for our purposes. The one thing that is important, particularly the genotype, is all the genes that an organism has. So if we study that on two different strains of bacteria, we might find that they are slightly different in terms of their genotype. There might be some mutation or loss of a gene or something like that. Whereas phenotype is all the expressed genes. What is the properties and appearance of an organism based on which of the genes it has that are expressed? So just because the genotype is one way, it doesn't mean that the phenotype will be necessarily 100% predictable. There are other factors that can influence which genes are expressed. This is just a diagram of the sequence of some related viruses from different parts of the world. We can see that some of the viruses from different areas, different regions, are very, very similar to each other in this particular part of their genome. It's just one part of the genome was sequenced. We can see this Egypt and France strains are identical within this stretch of base pairs that we see from the original genome, whereas other ones are a little bit different, but yet we see two other, at least, sets of related, very closely related viral strains, and some that are quite different. So studying genomics can help us identify the relatedness and possibly the history and origins of particular strains of viruses and DNA, or viruses and bacteria and so forth. So this is just a little analysis of some of those strains that we see in this in that table. There's a famous and very beautiful micrograph of the genome of an E. coli cell that's been disrupted, being broken open, so that the DNA, the genomic DNA, can escape. Now, isn't this interesting? It's an amazingly long piece of circular DNA that was all packaged up inside this bacterium with all the other tasks and whatever else is that thing's doing. It's able to transcribe and translate genes from this incredibly packed in genome. So it's just a miraculous thing, I think, to conceive of, to wrap your head around. Think of that whole thing being packed in there, and yet efficient things can happen. Another thing wants to replicate its genome, it can make an identical copy of this thing in relatively short order and cells can replicate every half an hour. So keep that in mind when we're looking at the actual size of that genome. Here's a little map of the E. coli genome, and some of the genes are shown in little colored blocks. So not all of the DNA is taken up in the recipes or the coding regions for particular proteins, but some are. And you see some of them listed down here. Genes involved in DNA replication and repair in case something happens by way of a mutation in the DNA. Lipid metabolism, carbohydrate metabolism, so all these different functional genes in the genome of the E. coli. Note the number of base pairs we're talking about. The left-handmost column place in these numbers is millions of base pairs. So we're talking about a 5 million, between 4 and 5 million base pairs of DNA in this double-stranded circle. So remember, every half an hour, those 4-something million base pairs can be accurately replicated in order to produce 2 daughter cells, each with an identical chromosome, nearly so. So what happens in terms of genomics? One thing that can happen is we can express genes, transcribe the sequence for making a protein out into a messenger RNA and then translate that into new proteins. So that's what's being shown here in this part of the diagram. Recombination can actually occur. We'll find out that genetic information between genomes, between chromosomes, can be swapped and shared. And here's another process happening we'll be talking about, where when a cell is about to divide, it will replicate its genome, make an identical copy of the genome, and we'll see how that's done, how bacteria can actually do that, which is somewhat similar to how it's done in animal cells or in eukaryotes. So, what exactly is DNA? And DNA is a polymer of nucleotides, nucleotide bases with hex or ribose sugars connected together in a long chain by phosphates. So let's take a look at it's easier to look at this in a picture. So here we have the bases, purine and pyrimidine bases, each one of which attached to a 5-carbon sugar, 1, 2, 3, 4, 5 carbon sugar, and then each one of the sugars, the ribose sugars, is connected by a phosphate. And so we have 2 different strands in a double helix of DNA. DNA forms a double helix, and the bases hydrogen bond together like little magnets, and they're selective. There's 4 nucleotides: adenine, thymine, cytosine, and guanine. That's what's represented by these colored boxes, those 4 nucleotides, and they pair in exactly the way shown here. Guanine pairs with cytosine, adenine pairs with thymine, and the hydrogen bond to connect together. So they're like little magnets. The backbones are very tightly attached. All these bonds are very, very durable bonds, but these hydrogen bonds can be undone. We can unzip, pull this whole thing apart, and then let it kind of snap back together like magnets, which is kind of cool. So we'll be thinking about that when we talk about unzipping and rezipping a double-stranded DNA. Another thing that's very important to note is there's directionality to this thing. Look at the ribose sugar. On carbon 3, there's a hydroxyl group on one free end of this chain. And then here's carbon 5. Carbon 5 has a phosphate bound to it. But when we're going to add, if you want to add a nucleotide base to this to a DNA chain to make it longer, it's going to be added on to the 5' end. It turns out that the enzymes that add bases add onto the 5' end. 5' means here's that carbon 5 with its phosphate. We're going to grab a new nucleotide base and take its 3' hydroxyl group, not from this chain, just a free one, and stick it on and elongate this chain. Again, the polymerase proteins that make DNA and RNA can only proceed to build in one direction, 5' to 3' it's called. So that's what's meant by that. We're talking about adding bases onto the phosphate that's attached to carbon 5, and we'll take a carbon 3 hydroxyl group and fasten it onto that phosphate. So here you see a similar kind of a process may be about to happen. If we unzip this double helix, free nucleotides can hydrogen bond, find their partners, and then an enzyme can come along and covalently attach them permanently onto the growing chain. And we can form from one chain, we can form two chains, we can unzip it and form an identical copy onto each of the original chains. That's exactly how DNA is replicated. It's called semi-conservative replication because always there's one original strand of DNA onto which a copy is made, and that forms a copy that can go into one daughter cell, and then the other original strand of DNA will then be used to make a copy, and so now we have a double helix to place in each cell. But each daughter cell has one strand that was originally present in the parent cell. Leads to a fairly high level of integrity in replication. So here we see again a polymerase adding a nucleotide base on in the 5' to 3' direction. You need a nucleotide triphosphate, or nucleoside triphosphate, to add on to that to form this adding chain. Maybe I said that backwards, 5' to 3'. Here we go, adding this. The 3 phosphates, the triphosphate is on the 5' carbon you see here. We're going to break off 2 phosphates. We're just going to release some energy and attach this base onto the growing chain, onto that 3' hydroxyl group. Okay, let's think about the actual process of replicating a double-stranded DNA. There's a problem inherent in the fact that the polymerase can only proceed in one direction, but the two strands of DNA are anti-parallel. They're facing opposite directions. I started to speak about that here and I didn't finish. If you look at the 3' and 5' ends that are labeled here, look at the position of the orientation of the riboses and their carbons. This ribose is leading with the 5' end. This ribose is leading with the 3' end. So the direction of the strands is opposite. So when we replicate the DNA, the polymerase can only go in one direction, and that's going to lead to a little tricky problem in order to be able to do this process. Let's take a look at a picture that will help us see what's going on. You do not need to memorize all these names of different enzymes that are involved. It's more of a shock and awe slide to go, wow, how complex is it, just even in a prokaryote to replicate the DNA, let alone in a eukaryote. So here we have a double-stranded DNA. Part of the circular chromosome is going to be unzipping, and here it's being unzipped by an enzyme. And we're going to start copying. Now, this strand here is oriented in such a way that the polymerase can just be building. As you unzip, the polymerase just keeps adding bases that are complementary, it said, meaning the A's and T's are going to hybridize, and the C's and G's are going to hybridize, and then this DNA polymerase is going to put those in there and fasten them together permanently. So now we have a double-stranded DNA. How about the other strand? The other strand is facing the opposite way. The DNA polymerase, if you're looking at this slide with me, can only go from left to right. That's the direction it can polymerize. So it can't just keep going along the way on the leading strand it can, because this lagging strand, the polymerase is heading the wrong way. So it keeps on having to have a little RNA primer produced by one enzyme, a couple of RNA molecules that are hybridizing with the nucleotide bases will be put in there, and the DNA polymerase will go from left to right for a while and build a little bit of DNA, and then back up as the unzipping continues. A new RNA primer will be put in there, and a new piece of DNA will be produced from left to right, and so forth. They keep on jumping back and making more and more of these little fragments. And then a different enzyme has to come along and degrade that RNA primer and fill in the gap and fasten everything together. So you see the problem now because of the directionality of the polymerase and the anti-parallel nature of the DNA strands. Those little pieces of DNA that are produced each time the DNA polymerase produces a segment, replicates a segment that has to jump back, they're called Okazaki fragments. So this little piece is an Okazaki fragment. There's another Okazaki fragment, and those will then be connected. That will fill in the gap here between them and fasten them together. And that's the nature of trying to replicate the lagging strand. And this happens throughout the entire circular chromosome. Just keep going around until we form two completed replicated strands of DNA. Here's another cartoon diagram showing. Here's the original chromosome of a bacterium, and then at the origin of replication, it's going to start to unzip and start copying, making two identical copies at the same time. Each new copy will have one of the original strands plus a new strand in it, and we're just going to work our way around this circle until the very, very end, and we get two identical replicated double-stranded chromosomes. An enzyme will come along and help them separate, and then they can be shipped off to the two different daughter cells. So that's kind of something about the way that replication happens, and just getting into the fun of understanding that problem of the directionality of DNA polymerase and the anti-parallel or opposite direction of the DNA strands. How about transcription? We want to make some protein in the cell. We need to transcribe or copy out the recipe for the sequence of amino acids into a messenger RNA, and that can be sent out onto the ribosomes to be translated. So a gene has the coding region. That's the information for where to put which amino acids to put in. And there's a promoter region, which is going to be regulating that gene. When is it on and when is it off? Not making any protein or messenger RNA for protein. So that's the promoter. There's a terminator sequence, so that let's say we have a 4.5 million base pair chromosome that has 30, 40, 50 genes on it. We only want to transcribe one gene. There's a stop codon, a terminator sequence in there to stop the replication. Shouldn't have said stop codon. I'm jumping ahead of myself. A terminator sequence so that the transcription will end, and we just copy out the gene of interest on that particular occasion when that promoter is turned on. So here we see a little cartoon showing a promoter. This is a specialized sequence within the genome to allow an RNA polymerase to stick down onto there and begin unzipping the double-stranded DNA. And then the RNA polymerase can start copying that naked strand of DNA with RNA. You see the RNA polymerase uses a different type of bases. DNA is deoxyribonuclease, so the ribose sugars are missing a hydroxyl group compared to RNAs. These are different nucleotide bases. You see that the thymine is replaced by uracil in the RNA. Anyway, for what it's worth, so we're going to copy out the sequence of DNA in RNA so we can ship out that RNA and make some protein out of it. And you can see that as the RNA polymerase moves from left to right in this diagram, you see this growing messenger RNA being produced, which has the exact same sequence as the DNA had. It's like making a copy of something. You want to preserve the original, and then you can send the copy out to go to work. So that's the messenger RNA being made that has the complementary sequence. The bases are pairing exactly the way they paired in the double-stranded DNA, only now we have an RNA that can be sent out separated from the DNA. Real quick, before we go on to looking what happens as far as translation, making the protein, in eukaryotes, this sequence on the DNA that's going to be copied into the RNA has extra segments in there, extra sequence in there that has nothing to do with the recipe for making the protein. The parts of this gene that are going to be used to directly translate into protein sequence is called the exon. There's multiple exons in this gene, but then there's some intervening sequences that are not going to be copied, used to encode the amino acid sequence in a protein. They're called introns. So when the messenger RNA is first produced, it's going to have exons and introns in it. Here's the transcript, exons and introns in there. Some specialized enzymes have to come along and cut out all the introns and splice the exons together into a contiguous strand. Now that's called messenger RNA. We start out with just a transcript. Now it's messenger RNA because we got rid of all the introns, and now we can actually use that to directly translate a protein. And in a eukaryote, that mRNA mRNA is going to go out of the nucleus into the cytoplasm to be translated. In prokaryotes, there are no introns. All of the sequence is contiguous in the chromosomes in the genome itself. All right, how in the world do we translate? Well, mRNA, every three base pairs in the bases in the RNA represent one codon, which defines an amino acid that will be used to put into a growing chain of amino acids. I don't know if I said that very clearly, but every three bases in the messenger RNA corresponds to one specific amino acid. The very first codon in the messenger RNA is AUG, and then there's three possibilities for a stop codon. So that again, just like when we had a terminator sequence in transcription, we have a stop codon to end the protein. When we get to the end of the protein sequence, you'll see a stop codon. RNA polymerase will fall off and stop translating. So again, we're talking about this translation process, talking about transcribing the DNA into a messenger RNA, and now we're going to translate it into a protein. So let's see how that process works. Well, out of three, there are 64 sense codons, but only 20 amino acids that they encode for. And it turns out that the genetic code is degenerate. A better word for that might be redundant. There's redundancy in the three base codons that are used to select RNAs. Transfer RNAs are separate little RNAs that will actually hold the amino acids and bring them to the ribosome to translate, and they have three base codons that are the opposite of the codons in the messenger RNA. They're complementary, to those bases, and let me show you in a picture. It's a little easier. So every three bases in the messenger RNA calls for a specific amino acid. A particular transfer RNA will come along and bind to these three bases, and with that transfer RNA will be an amino acid. In this case, if it was uracil, or uracil cytosine, a transfer RNA with phenylalanine would come along, and that would be added to the growing chain. So as you see, let's look over here at this serine selection. The sequence U and C always gives rise to a serine. Doesn't matter what's in that third position of the codon, U, C, A, or G. There's the four different nucleotides that could be there. You'll always call for a serine. So that's what we mean by redundancy or degeneracy. So you see that out of this entire table of 64, only 20 amino acids are called for. So it's interesting because let's say we had a mutation in this third position. We changed the base by accident in the genome. It wouldn't make any difference. Serine would still be called for. In order for a mutation to occur in that changes the amino acid sequence, it would have to be in one of these first two positions in the codon. Again, there's some stop codons. If UAA is present, the RNA polymerase will fall off the strand. Here's the AUG start codon. It encodes for methionine. So, how efficient is this process? Here's our genome, our gene in the genomic DNA in the chromosome, and here's the RNA polymerase unzipping and forming a RNA replicant. You can't hardly see it here, but it's dangling down the RNA as it grows longer. As the ribosome moves from right to left in this picture, the messenger RNA is getting longer and longer, but there's no need to wait around for it to be finished. As soon as that messenger RNA begins to stick out from the RNA polymerase, a ribosome, the little red things are ribosomes, assemble right onto there and start producing or translating a peptide, a protein from it. And so by the time this messenger RNA gets to any impressive length, look at all the ribosomes that are packed on there, just producing protein right away in rapid form. Pretty impressive and efficient and cool. All right, how about the ribosome itself? It's got two subunits. we said that bacteria have the 70S ribosome, and it has these two subunits which at the AUG start codon will assemble together on the messenger RNA like a little machine, and transfer RNAs will start to come in. So the first transfer RNA that recognizes the AUG will assemble together with the ribosome and form the starting equipment. Then the next transfer RNA that recognizes or is complementary to the next codon, the next three bases in the messenger RNA, will come in and hydrogen bond on there and then attach. Methionine will then be attached onto that leucine. And so we see what's happening. Once we get that second transfer RNA into this so-called A site of the ribosome, we're going to take an enzyme that will swap this methionine over onto the leucine. And then that leucine holding a transfer RNA is going to shift to the left into the P site. And another transfer RNA is going to come in there and bring its amino acid down. And then the enzyme is going to come and transfer the leucine with its methionine over onto the glycine, then onto the phenylalanine. We'll just keep on doing it. It keeps shifting. The messenger RNA with its transfer RNAs keeps shifting to the left, and the leftmost one on the E site keeps getting shucked off, and a new one keeps coming in into the A site, and then adding the growing peptide chain onto the amino acid that's on the transfer RNA in the A site, and so forth. And so we have this polypeptide forming, emerging from the ribosome as the messenger RNA is shuttling through there. Incidentally, this arrow looks confusing. It's showing you the direction of movement of the ribosome along the messenger RNA. The messenger RNA is going from right to left, as the ribosome is going from left to right. So otherwise that might be a little confusing, I thought. So we're going to wind up with a polypeptide chain until we come to a stop codon. We come to a stop codon, then the ribosome is just going to come apart and leave us with this free protein, brand new protein that's been translated. All right. Let's look at regulation of this process. What determines when that gene is going to be doing all that transcription and translation of a particular gene? Constitutive genes are ones that are always on. They're always producing their product. Some so-called housekeeping genes in the cell just keep on making their product. It keeps getting degraded. It's just always working. But oftentimes, genes are only turned on when it's appropriate, so we don't waste energy and resources in the cell. And in bacteria, some genes, the way they're arranged, and we'll see there's a special name for them, are inducible. They're normally off, then we can turn them on at the appropriate time. There's other genes that are normally on, we can shut them off at appropriate times when we no longer want them to be working. And there's some, and there's just an example of a repression being produced by a catabolite that's building up, and it's going to block the production, presumably, of an enzyme that breaks down that product. All right, so in order to discuss this just for fun, this is a really fascinating topic that's, we'll get into some very rudiments of how bacterial genes are controlled. And the bacterial genes are organized into a construction called an operon. There's structural genes. That means that's the coding region for actually producing the messenger RNA that can be made into a protein, translated into a protein. So it's meant by structural genes. It's the coding sequence. Then we have an operator to which an inhibitor can bind, a repressor can bind in this spot here. And then here is a promoter where we can get an RNA polymerase to attach on there and start going down and transcribing this gene. So here's a picture of an operon, an inducible operon. It's normally off because there's a repressor protein attached to the operator site right here. And so the RNA polymerase can't proceed, it's blocked. And so that thing will just sit there idling and not doing anything at this time. Why is that happening? Because there's a gene upstream of the structural genes that produces this repressor protein, and it's ready always to bind into the operator to stop this thing from going. So here's the spot where the regulatory gene is that produces this repressor. So it's called an inducible operon because if a particular substrate comes along that would be metabolized by some enzymes produced by the structural region, the structural RNA up or DNA up here. So if that particular substrate comes along, like lactose in this case, we might want some genes to metabolize lactose. And so if lactose comes along, allolactose binds to this repressor that we saw, it changes its conformation, it no longer binds to the operator. And if that's the case, then the RNA polymerase can whiz right down and make messenger RNA and translate it into some genes, beta-galactosidase, for example, that will help us metabolize lactose. So is there lactose present? Yes, good. Let's make some enzymes to metabolize it. If not, no. The lactose, the lac operon will be turned off by this repressor. So that's an inducible operon. How about a repressible operon? A repressible operon is normally on. So it has the same components, structural genes, an operator and a promoter, that's the control region, and then a regulatory gene. What's going on in this case? In this case, the repressor protein is normally inactive. It can't bind to the operator, so this thing is going gangbusters and making messenger RNA and proteins. In this case, it's for synthesis of tryptophan. That's one amino acid that's needed by the cell to make all of its proteins. And so if we don't have enough tryptophan, we're going to be making some by the products of the structural gene here. However, as tryptophan becomes prominent or prevalent in the cell, we don't want to keep on making more tryptophan when there's already plenty. Tryptophan binds to the repressor and changes its conformation, so it does bind to the operator. So when tryptophan begins to build up, we have a negative feedback effect that's going to block the transcription of more enzymes to synthesize tryptophan. So you can see how we're going to be efficient. We're not going to keep making tryptophan when we've got enough tryptophan already by this repressible operon. One step of complexity further. Here's some bacteria. And you can see that when this particular bacteria has the capability of fermenting either glucose or lactose, or metabolizing glucose or lactose. When it's metabolizing glucose, it's able to grow more quickly than when metabolizing lactose. There's more energy produced more efficient. But at some point, let's say both of those substrates were present. When both substrates are present, the bacteria will grow on glucose at the fast rate. So here we have a mixture of both lactose and glucose, and the cells are using only the glucose, growing very, very fast. But then when the glucose runs out, at this point right here, then there's a little bit of a lag, and then lactose becomes the substrate that's metabolized in order to make this thing still work. How does all that work? Well, when glucose is absent, then cyclic AMP levels build up in the cell. When there's plenty of glucose, the cyclic AMP level is low, the second messenger, and when the glucose is absent, then cyclic AMP levels go up. Let's see how that affects the picture here. So when cyclic AMP is present, it binds to this cap protein, and the cap protein can come and bind to the promoter and activate transcription, and will produce lactose metabolizing genes. When there's plenty of glucose present, that is, when there's lactose present, then we won't make the genes for producing glucose metabolism. If the glucose is scarce, there'll be high level of cyclic AMP, activates the cap, and will produce large amounts of mRNA for lactose production. So if glucose is scarce, cyclic AMP, as I was saying at the start, is high, will activate the cap, and will make lactose metabolizing proteins. But if there were plenty of glucose around, then there would not be any cyclic AMP, and the cap protein would be inactive and would not waste energy making these lactose metabolizing enzymes. And that's why when glucose is present, the cell, so down here, when glucose and lactose are present, the cells will strictly metabolize glucose, because we're down here in this picture, there's no active cap. We can't turn on you can't turn on the LAC-Z gene, so quote unquote the lactose metabolizing enzymes. Whew. Okay, Epigenetics, what's the deal with that? Well, genetics, if we just look at the correspondence between genes and their gene products, there could be a correspondence between genotype and phenotype. If the gene's there, it gets turned on. Now we have a particular phenotype. Well, it turns out there's something else that can affect phenotype other than is the gene present or not, because some genes can be turned on and off by methylation. We saw some ways genes can be turned on and off by repressors. Well, genes can also be turned on and off by methylation, adding a carbon with its hydrogens onto a location in the genome that prevents transcription of the message. So methylation of genes is a way to control which genes are on and off in a complex genome. It's not permanent. It's not part of the actual coding. It's not part of the code. It's not part of the genomic makeup of the organism, but it definitely affects the phenotype because it determines which genes are actually expressed, which proteins the cell is making. So biofilms, when are biofilms produced? When there's a large number of bacteria accumulating, and they'll start secreting some signaling molecules, which will ultimately turn off some genes, turn on some other genes to make the biofilm. And those kinds of controls are called epigenetic controls, and they can actually be inherited. If there's a bunch of different genes that are methylated in a parent cell, and then those genes are replicated, the genome is replicated for the daughter cells, the methylation can be carried over. And in fact, certain traits in higher animals can actually be carried over in offspring because of patterns of methylation of the genome that has nothing to do with the actual genes that are present, interestingly. All right, so I'll come back in a little bit and look at another part of this chapter, and we'll learn about how bacteria can swap genetic information within the same generation, which is kind of a unfortunate little trick, because a disease-causing gene in one bacterial species can be transferred to another if they're both present in the body at the same time.